All right. Hey, everybody, what's up? How's it going? Class number 5, ready to go. Uh, how's everything been going? Have people been doing the experiments? Anybody send anything out for sequencing yet? Hey, everybody, what's up? How's it going? Class number 5. Sorry about that. Yeah, has anybody sent anything out for sequencing yet? Got any results back? Still waiting on that to happen. Anybody purified any plasmid? That is going to be essential in completing the next part. You're going to have to purify some plasmid. So get on it. Purify your plasmids. You're going to need to do that. Yeah. So today what we are going to talk about is after you purify your plasmid, what you're going to do. Now, one of the big points of this class is not just to purify plasmid DNA and sequence plasmid DNA, but it's also to learn how to combine different pieces of plasmid DNA so that you can create new custom plasmids. So we're going to focus on that. Now, generally, nowadays, what you can do is you can pay a company a few $100s, maybe like, I don't know, $200-300, and they will put a custom piece of DNA inside a plasmid for you, where you tell them. But it costs a few $100. The other way is you can find a plasmid that already has the piece of DNA you want if it's common, and you could cut it out and you could put it in the plasmid you want. That is what we are going to talk about. Now, this way is much, much, much less expensive. You can do it for the cost of the plasmid purification and the restriction enzymes and the sequencing. If it works out correctly, generally it'll cost you, total about $10 or something. Obviously, these things are complicated, so they don't always work correctly the first time. But that's what this class is about. It's learning how to do these things so that you have more of an intuition for what could go wrong, what might go wrong. So we're going to move into this next part and talk about restriction enzymes, what's going to happen, and all that fancy stuff. So if we go to our Google Classroom and let's find our README for week 5. Restriction digest ligation transformation. So there should be a protocol in there for week 5 in the README that describes exactly what you're going to do. Describes how to use restriction enzymes and how to stitch things together. All right? So we are going to use 2 restriction enzymes. We are going to use XHO1 and XBA1. First, what we can do is let's look at the SNAP gene. All right, so what I'll do is I'll pull up SNAP gene. Let's open recent file. Let's open, do we have Pdog? Let's open Pdog. All right. Let me pull that up on the screen. Let's see, display capture. Oh, all right. Pdog. All right. So this plasmid is a little flipped around, but we'll go with it. So what we want to do is we want to create cuts at the XBA1 and the XHO1. Right? These are the two sites that what happens is if you purify this Pdog plasmid, mix it with XBA1, mix it with XHO1, everything in between will be cut out. So we're talking about all this stuff. All that will be cut out. So we'll have two separate pieces of DNA, right? And now what we want to do is what's actually in here. You can't see it in this plasmid map because this plasmid map doesn't contain the full Pdog. Pdog actually has red fluorescent protein in between the XHO1 and XBA1. Where is XBA1? Enzymes, XBA1. Oh, nice. Cuts out that whole section right there. All right. So actually there's two XBA1 sites in Pdog. The other one is this whole lambda repressor. So we're actually going to cut out this whole lambda repressor. We're going to cut out the other XBA1 site, and we're going to cut out the XHO1 site, right? Because it's going to make a cut at both of those places. So it'll cut out all this stuff, which will just mostly be the lambda repressor and the DS red, which is good for us. We don't want the lambda repressor there necessarily. That means we should get a constitutively active whatever we stick in. So what we want to stick in is we want to stick in a green fluorescent protein. Let's see, open Google Drive. Let's go to plasmids. Where's plasmids? Plasmid. And then, let's see, GFP. All right. Now if we find the same XHO1 and XBA1, so generally when you're looking for restriction enzyme sites, you're looking for restriction enzyme sites on the outside of where your gene you're trying to cut out. So this XBA1 site, right? We're trying to cut out the green fluorescent protein. That's before the beginning of it. And then the XHO1 site is at the end of it, right? So what this should do, if we use the XHO1 and XBA1 sites, is it should cut out the whole green fluorescent protein. So in the Pdog plasmid, we should cut out the whole RFP protein. And in the GFP plasmid, we should cut out the whole GFP protein DNA sequence. So what we should have after this is the RFP and GFP DNA sequences and the plasmid backbones, right? So let's try to visualize this. All right. Let's see if we can do this. All right. So we have so what we have here is this is Pdog, and in Pdog we have XHO1 and XBA1. This is GFP, and what we have is XBA1 and XHO1. So most plasmids are kind of designed similarly, where they'll have common restriction enzymes that are before and after the gene. This helps when you're trying to move things between plasmids, right? So now what happens is we purify our plasmids. So step one, purify plasmid. All right. Now once you have the plasmid, what you want to do is you send it off for sequencing, but then what we'll do is we will perform a restriction digest. All right. So basically what we're doing in this case is we take Pdog and we add XBA1 enzyme and XHO1 enzyme. Now when we mix all these together, what happens is, right, it cuts it out of the plasmid. Come on, why can't I? Can I make this bigger? No. So bogus. Cuts it out of the plasmid. And then we have this, which is the RFP gene, right? Now the RFP gene still has the XBA1 site on the end and XHO1 site on the end. And now remember, when restriction enzymes cut, if we have two pieces of DNA with our base pairs, now when restriction enzymes cut, they cut in what they call an overhanging pattern. So it'll cut like this. And so what we end up with is we end up with a piece of DNA that has an overhang, right? So there's no top strand on it. That's what happens. You get an overhang on both sides where the restriction enzymes cut. Now, not all restriction enzymes have these overhangs, but you know, most of them, most of the ones people use most often do. Now, there are other types of restriction enzyme cloning or engineering that use enzymes that don't have overhangs, but we're not going to focus on that in this class. Now, why this is helpful is because it gives this piece of DNA a directionality, right? So you can imagine if, say, we had well, that didn't work out well. Why isn't that working out? Say if right here we had two XHO1 sites, right? We had one here and here. That means that there's two different orientations that the DNA can be put in, right? So it can be put in with the XHO1 site here or this XHO1 site there, if this is also XHO1. So using a singular restriction enzyme on both sides, it makes it more difficult to insert the piece of DNA in the correct orientation. So it's good to have two different restriction enzymes. All right. Let's clear all this stuff up. All right. So what should happen if we mix Pdog and GFP with the restriction enzymes, we should have Pdog backbone. We should have GFP backbone. And then we should have RFP and GFP, right? Now this is perfect. All we need to do then is get the GFP to go into the Pdog backbone or the RFP to go in the GFP backbone. Now, how can we make that work? Well, remember that these plasmids have antibiotic selection. The GFP has ampicillin antibiotic selection, and the RFP has kanamycin antibiotic selection, right? So basically, what's going to happen is after we do the restriction digest, we will use a DNA ligase. This DNA ligase, what it does is it creates bonds on the DNA backbone. The bonds created here will connect whatever's near. So if the RFP is near the Pdog backbone, the RFP will go back in. If the GFP is near the GFP backbone, it will go back in. But if the GFP is near the Pdog backbone or the RFP is near the GFP backbone, then it will go in. So what you'll have is you can have the Pdog with a GFP and the GFP backbone with an RFP. Now, how do we make this happen? Right? All right. Do we have any questions so far? How's everything going? Oh, wrong screen. Let's see. All right. Any questions? Any questions? Any questions on what I'm talking about? Does it make sense? So you can move things between plasmids when there are similar restriction enzyme sites. There are ways to do it when there aren't similar restriction enzyme sites, but generally you want to have similar restriction enzyme sites. It's just easier all around. Any questions? Any questions? All right. Doesn't look like there's any questions right now. So we'll go back and talk a little more. All right. All right. Control ++. Brushes. All right. So let's erase this. All right. So in our tube, we added the DNA ligase, and this connects the GFP in some of the Pdog plasmids and the RFP in some of the GFP plasmids. And the way we can tell this is by the antibiotic selection, right? So what we do is we do a DNA transformation. So when you do your DNA transformation, make sure you do a positive control of some of your plasmid. So some of the plasmid you purified, do a positive controlled DNA transformation. So say take your GFP plasmid, purify it, do a transformation, and make sure that your transformations work before you try to transform your ligations. Because ligations have a lower probability of working because there's going to be less plasmid in there. So step 4, do a GFP transformation, right? Make sure you get that, make sure it works. Make sure you can see GFP colonies, right? So if you get there and you see GFP colonies, then the next step is to do a transformation of your ligation, right? So your ligation, it can be frozen at almost any time. So after you purify your plasmid, you can freeze it. After you perform the restriction digest, first heat to nature, and then freeze. So what heat to nature is it is putting the solution at 65°C for like 20 to 30 minutes. And what happens is it kills all the restriction enzymes. So the restriction enzymes can no longer cut DNA, which is good. Because in the next step in the DNA ligation, we don't want the restriction enzymes to cut all the DNA we ligate. So I would do the heat denaturation step before I freeze, just because you'll still, I guess you can wait, but like I would do it before if at all possible. Do the heat denaturation and then freeze. After you do your DNA ligation, you can freeze it, right? So after all these steps, it's possible. So you can, after you do your DNA ligation, you can store it in the freezer, use some of your purified plasmid from before to do a GFP transformation, right? And after you have all that working, then go ahead and start working on trying to get these plasmids transformed. Now, what this is going to happen is we're going to transform these, and then we're going to plate it on kanamycin and ampicillin. Now, if we see red, so if we see little red dots on the kanamycin, we know that it didn't work, right? Because Pdog should, in our engineering, should have GFP. Now, what you're going to see is you're probably going to see red dots on the kanamycin. That's okay. But what you want to look for is you want to look for the few green dots that are going to be on there, right? So it's probably going to be majority of red dots and some green dots. You might even only have like 1 or 2 actual green dots, right? But that's okay. The number of Pdogs that have Pdog or didn't get cut are going to be higher than the probability of the GFP piece of DNA coming into the Pdog, getting ligated, and then transforming properly, right? So the GFP on the kanamycin means that the GFP made it right into the Pdog plasmid, because in the GFP plasmid, it has ampicillin resistance. So none of the green colonies should survive in kanamycin because it can only survive on ampicillin. So on the GFP plasmid, what you should see is you should probably see a bunch of green dots and then a few red dots where the RFP made it into the GFP plasmid, the RFP gene. So it's going to be the opposite of this one, right? Because the Pdog plasmid, when stuck in bacteria, it should it, it shouldn't be able to survive on ampicillin media. Not at all. It should be able to survive on kanamycin media, but not ampicillin. So the red dots should be from GFP plasmid with RFP DNA in it. Make sense? Does this all make sense to everybody? So yeah, so Kent, could utilizing two selective factors result in the uptake of both the GFP and RFP? It probably won't be into one plasmid backbone, right? So remember what we have. Let's go back and let's look at this really quick. All right. All right. Let's clear out all this. All right. Oh gosh, what happened? Really weird. Paint. All right. All right, so remember in the plasmid, what we have is we have the XBA1 site and the XHO1 site. So when this comes out, what you're going to have is you're going to have RFP with an XBA1 site and an XHO1 site, right? Now the GFP is similarly going to have an XBA1 site and an XHO1 site. So this XBA1 site can't connect to this XHO1 site, right? So if the half of the GFP bound to here and half the RP bound to here, it wouldn't create a full circle. So it probably wouldn't transform or be really transformable. So we need both the XBA1 site on that side and the XHO1 site on this side in order for the gene to get in and get ligated. So there's only a possibility of exactly one gene, in this case being in the plasmid, unless something crazy happens. And sometimes crazy shit happens, but molecular biology is just a game of statistics. But it's generally not going to happen. We're only going to see one gene in the plasmid at a time. Does that make sense? So like we should only get one. Now, if you're trying to select both GFP and or both ampicillin and kanamycin, most likely you're probably going to get not much of either. You probably can try to get bacteria that have both kanamycin and ampicillin. If you say just transform the GFP plasmid and the Pdog plasmid by themselves. But generally, if you try to transform two of the cut plasmids, it's probably not going to happen. You have to understand that when you do like restriction enzyme cutting and stuff like that, the probability of the transformations working go down because you're cutting up the plasmids, right? You're already destroying them. And you're probably using less DNA for the transformation than you'd normally use for a transformation. So what happens is your probability of getting the transformation to work is less when you're doing the restriction digest. So being able to get both of them in while possible, it's not as likely. It's possible, just not as likely. All right. Any other questions for right now? Any other questions? Hey, Joe, how did your sequencing come back? Did you get the sequencing back and did it work well? Um, Joe Dart, I'm interested. So, what what we have is you didn't get it back yet? Okay, cool. Let us know when you do. Um, all right. So here's the things that we need, right? Think about the flow of the experiment. 1, make sure your transformations work, right? Especially if you've never done transformation before, this should be your goal. Make sure your transformations work. So um, if you're working with a plasmid that you purify, it's also going to be more difficult than, say, the plasmid you get from an Odin kit. Because the plasmid you probably get in an Odin kit is higher concentration. So you want to make sure you can do a transformation properly, right? So first try to do a transformation. Grow up some of that DH5 alpha bacteria and do a transformation with your purified plasmid and try it until you get it to work. Sometimes what you might need to do is increase, my handwriting's atrocious, isn't it? Increase the volume of plasmid. So what I would try to do is for my transformation is start off with 10 microliters. And if it doesn't work, then try 20. That might take up all of your plasmid that you have, right? Because that's 30 microliters. And usually you purify, you elute with like 50 microliters or so. So you might have to do it over again. But the transformation is highly concentration dependent. So if you have a low concentration, you're going to need to use more, right? The higher concentration, the less you need to use, right? Because by increasing the volume, all you're theoretically doing is increasing the concentration. But yeah, do it until you get this correct. Because if you don't get the transformation correct, you're not going to be able to do the next steps. Then what you need to do is you need to purify Pdog and GFP. Run, a little bit out on a gel, make sure you have some there, and then do a restriction enzyme digest of Pdog and GFP, right? So another good thing is is when you grow up these cultures, so if you grow up the Pdog and GFP cultures for mini prep, you can also freeze them, right? So once you spin them down, you can freeze them. So there are a lot of places where you can just stop in this process, right? Don't don't necessarily try to do it all at once. Don't try to do it exhausted either. Try to make sure you do a good job on this because it is like a skill that you have to learn. And if you're tired and exhausted, it's generally going to be hard. So, like do it a little bit at a time. That's okay. Um, for this next part of the project, we're going to have 3 weeks. The last 3 weeks of this class will be trying to accomplish this project, taking everything you've learned and putting it to use. Right? Then he denature the restriction enzymes, ligation to try to switch the GFP and RFP, transformation, and then plasmid purify of your colonies. So what should happen is you should get a Petri plate, right? And it should have little red or green colonies. What you're going to do is you're going to take one single colony and you're going to dip it in some LB, right? You know how you use an inoculation loop to grow up your bacteria for plasmid purification? Well, you're going to do the same thing, except instead of sticking it in a bacterial stab, you're going to stick it in a small colony that grows on your plate. So take one colony at a time. A colony is just a little circle of bacteria. So take one colony, grow it up in LB with the proper antibiotic for whatever antibiotic you're using. So purify that plasmid. And then finally, what you're going to do is you're going to send it for sequencing. Right? So this is a lot of stuff to do. So transformation, if you already have plasmid, this will probably take like a few hours worth of work. So maybe like 1 to 2 hours work. And then you're going to have to let it sit overnight to wait for your bacteria to grow. Purifying Pdog and GFP, you're going to have to let the cultures grow overnight. And then it's probably going to be 1 hour to purify the plasmids. 1 to 2 hours because you're probably going to run a gel also. Restriction enzyme digest, you're going to let the digest go for about 1 hour. Take a little bit to set up, so maybe 1.5 hours. Heat, deactivate, that'll be another 30 minutes. Ligation, depending on which way you do it, it could be 1 hour to overnight. The transformation, right? You're going to have to grow up some fresh DH5 alpha for that. So you'll have to start that the night before. Try to use fresh DH5 alpha for your transformation, as fresh as possible, right? And then your transformation is going to take 1 to 2 hours plus overnight to grow the plate, right? So we're talking a lot of work here, but this is the workflow that you're going to need to do normally in genetic engineering, molecular biology. This is what you would do if you get a job in a lab. This is the kind of stuff you're going to have to do. It's just you find a process like this, right, that works, and then you use it to engineer different things. Now, one thing I want to go over real quick before we go, well, maybe we could go over that next week, how to order custom DNA Um, how to order custom DNA if you're trying to do insert custom DNA into a plasmid instead of just doing a restriction enzyme swap between two plasmids. That's something that we can talk about next week. Um, in the meantime, you guys need to catch up. Um, you need to get back on this. Get those plasmid purifications going. If you have any trouble with the plasmid purifications, also let me know. I'm here to help. There's no stupid questions. Seriously, no stupid questions. um, yeah. So Tom asks, there are some bacteria that are proficient at replicating plasmids and others protein synthesis. Yes. So generally people use DH5 alpha bacteria for replicating plasmids. This is because they lack some recombinases. And so the plasmids survive better. Now, how much better? It's unclear. But the two common strains are DH5 alpha and BL21. Now, BL21 has been infected with a T7 polymerase. So it has a T7 polymerase in this genome. Now, this is a viral polymerase, and this viral polymerase is very efficient. It works really fast, right? So it can make lots of mRNA, which can make lots of protein, right? And so generally BL21 is used for making protein, and DH5 alpha is used for making DNA. You can purify DNA from BL21, and you can purify protein from DH5 alpha. It's totally possible, but generally this is the way it goes. So when you're doing all this cloning stuff, you're trying, right? All molecular biology is basically trying to push the statistics in your favor. What you want is you want to have the highest probability that your experiment works by using things that'll help you out. So you want to try to use DH5 alpha when you're doing all this cloning and stuff like that. No, so it's hard to necessarily overuse a selective antibiotic. Sometimes it's good to overuse if you think there might be contamination or something like that. So, if you use, say, 2 times the amount of kanamycin that is normal working concentration, or 2 times the amount of ampicillin that's normal working concentration, a bacteria with a plasmid should still survive. And it shouldn't necessarily have a negative effect on the bacteria, at least in my empirical experience. I have not seen it have a negative effect. And generally, when I'm messing around and I'm not really being sterile, I usually add more antibiotic just to be safe. It's okay, right? You're not contributing to antibiotic resistance in the world. The amount of antibiotics used in science is like 1/1 billionth of the amount of antibiotics used in agriculture. So don't worry too much about that stuff. Hey, Leslie, the 201, the human cell engineering class. If you're interested, sign up for the human cell engineering class. It's going to be awesome. So the stuff will probably start to ship in the next week or two. The microscope should work with any computer that has a USB plug, basically. So I wouldn't worry too much about it. But your stuff should start to ship in the next week or two. The thing is, with the human cells that we're sending, generally human cells are a lot more finicky, and you can't really store them well. So you kind of have to ship them really close to the time that the class is going to start, uh, so that people can work with them. So they're you generally with human cells, you can't really store them out in say the refrigerator and they'll last for a month. Um, you have to store them at room temperature and they're constantly growing while you're storing them. So um, yeah, you want to keep them as fresh as possible. So expect human cells, around the time of the class. Um, Leslie, what you can do is you could download the software. I think on the item description there is a link to the software for the microscope, so you can download the software for the microscope and see if it's compatible. SnapVolt, are these multi-copy plasmids? Yes. Almost every plasmid in bacteria is going to be a multi-copy. Um, in yeast, you can have singular copy plasmids. I'm not 100% certain if you can have single copy plasmids in mammalian cells. Um, but yeah, in bacteria, they're always multi copy. No, Leslie, actually, you can't store human cells generally at minus 80. I think it's something like you have to use liquid nitrogen. Um, so generally, what we teach people for the class, the human cell class, sorry to get off topic, is you keep the culture going. You just keep the culture going constantly. You keep it at room temp, and when you're trying to let it become a little bit more stagnant, and you just split your cells every so often, right? If you want to continue to working with them, you split your cells every month or two. Or every couple weeks, and you should be able to keep a constant culture going for a little while. Yeah. All right. Any more questions about the class? Any more questions about 102 class, not 201, which is coming up in the future? Any more questions? Anybody? Let me know. All right. So everybody get going on your experiments. We'll meet back next week, and you know, I'm here to answer your questions. Feel free to ask me any questions you have, right? Any questions you have about your experiments. If you're having trouble, if you're struggling with anything, just let me know. Um, I'm here to help you get through the experiment. Like I said, there's no stupid questions at all. Not at all. No matter what the question is, send it my way, and I'll do my best to answer it. All right? Otherwise, I will see you all next week. Keep on hacking them biologies.